We are now going to take a look at the first stage of mitosis, which is known as prophase. Prophase, it kind of starts off looking like this, which is what we ended with in G2, the last part of interphase. However, in mitosis, we need to make two cells from this one cell, and that's kind of what this white yarn kind of represents. It kind of represents the cell. So this would be the boundaries of a cell. So I'm inside the cell, this would be the cytoplasm in here, and then outside the cell, all right? So we need to take this cell and turn it into two cells. So obviously, in order to do that, we need to get some of this DNA over to one side and some of the DNA over to the other side of the cell so that we can then divide the cell in half, which is exactly why we made a copy of the DNA back in the S phase. There are now two strands of this yellow string, and I need to get one of those strands over here and one of those strands over here. Now, the reason why we need to separate these two strands of yellow string is because they contain certain genes on them. For example, maybe these are cells of your eye, and maybe your eye is green in color. Well, that trait, green, it might be linked to this yellow strand of DNA. So I need to make sure that I get green eyes over here, but also green eyes over here, because the two cells, they do need to be identical. If they are not identical, that is a major, major problem, and that can lead to all sorts of issues, as you may start to gather from previous discussions. Now, something happens to the DNA here, because right now this is a hot mess. As I mentioned previously, Christmas lights trying to untangle those. So how do we organize this so that when it's time to split up all of these strands of DNA, we can do so easily and effectively? And keep in mind, while I just have 4 strands of string here, in every single cell of our body at this point, we would have 96 strands, or 92 strands. That's a lot of string. So what we do is we take these loose, stringy strands of DNA and we wrap them around proteins called histones. I'm using a straw to replicate what's going on with a histone. And I'm going to use one of these yellow strands to kind of mimic the process. So just for the sake of space, I'm going to take one yellow out, get rid of those 3. And now DNA will essentially just start to wrap around this protein. Now notice as this process happens, what is happening to the DNA as this is occurring. So it's a nice long strand, and it's just going to start to wrap around this histone protein. Wrap, wrap, wrap. And notice how tight this is starting to become. Wrap, wrap, wrap. Nice and tight, almost done. And there, I now have my DNA wrapped around the histone. Just take the end here so that it stays. There. Now what I have is not DNA. It's no longer called that at this point. Now I have a chromosome. So here's a chromosome, and all the other strands are also going to wrap around, forming chromosomes as well. I've already done those to kind of speed up the process. So again, here's my kind of tannish one. Here's my yellow one. So now I have 4 chromosomes. And keep in mind, the yellow ones, they are identical, green eyes, green eyes. But these ones are also going to be identical. Maybe these will determine, height. So maybe a tall person. So this will also be a tall person. So now I have those. At this point, though, the physical appearance of the chromosomes, it does change. So chromosomes are made up of DNA, but it's made up of DNA that's now wrapped around a histone. So I have my chromosomes here. Chromosomes, at this point, start to take on an X-shaped appearance. Start to take on an X-shaped appearance. Now, here we have chromosomes, but here we also have chromosomes. This is a duplicated chromosome, as is this one, whereas these ones are single chromosomes. And because these two single chromosomes are exactly the same, they are said to be sister chromatids. So one duplicated chromosome is made up of two sister chromatids. We need to get one of these sister chromatids over to this side, and the other one over to this side, and same thing with this one. So, right now, because we're in prophase, these things are just kind of scattered about the cell, and keep in mind I just have 2, but in your bodies you would have 46. We now start to see the appearance of the formation of the spindle and the centrioles. These green X's, which are straws, represent my centrioles, and the brown thread here represents my spindle. And what starts to happen during prophase, first off, the DNA condenses. We get our duplicated chromosomes appearing, but we also start to see the centrioles and spindle begin to form, and the centrioles, they start to make their way to opposite sides of the cell a little bit. And that's prophase.